This is the cover slide, a small picture of adeno-associated virus AAV. This slide shows the entry of AAV into cell. After delivery into the human body, AAV first evades the human immune system and antibodies and reaches the target cell. By receptor binding, then AAV enters the cell cytoplasm through endosome and is transported by late endosome up to the nuclear membrane. It escapes from the late endosome and cellular degradation with proteasome and reaches the nuclear membrane. By transduction, AAV enters the cell nucleus. The genetic material is released single-stranded. The genetic material is converted to double-strand nature by cellular machinery. Some double-stranded AAV exists in cell nucleus as circular episomes, while the majority of AAV integrates by fusing itself at a highly specific site on human chromosome 19 by forming a concatemer. This slide is a detailed slide showing the behavior of AAV once it enters the cell nucleus, once again illustrating AAV's integration onto a highly specific site on human chromosome 19. This is a genetic locus map of human chromosome 19, which indicates all the human diseases and human traits with which human chromosome 19 is associated. Once AAV/AAV2 reaches the cell nucleus, it can go to one of two pathways. In the absence of a helper virus, AAV/AAV2 integrates as mentioned above on a specific site on human chromosome 19 via the latency pathway. In the presence of a helper virus such as adenovirus, AAV follows the productive pathway by replicating and forming very small AAV virion particles. The point here to understand is that AAV by itself, in the absence of a helper virus, is a replication incompetent virus. When AAV is delivered to the human body, we are not capitalizing on delivering a small amount of AAV and hoping for it to replicate and grow within the body, but rather we are delivering high concentration / adequate dosage of virus which will eventually follow the latency pathway. Another detailed slide indicating the behavior of AAV. As mentioned previously, single-stranded AAV is converted to double-stranded AAV by cellular machinery in the nucleus. Some AAV remain as episomes while other AAV integrate onto a specific site on human chromosome 19. Here is a slide showing the site on human chromosome 19 with AAV-S1 or Q13.3-QTER, onto which AAV integrates itself. AAV/AAV2 genome map. Understanding this is important. Wild-type AAV contains the rep, replication, and cap capsid gene. The rep gene codes for 4 proteins and the cap gene codes for 3 proteins. In recombinant AAV, the rep and cap gene are removed and supplied in trans. The therapeutic gene of interest is then inserted into the empty space, thereby forming the recombinant therapeutic AAV, now called RAAV. Recombinant AAV also behaves in the cell nucleus just like wild-type AAV. In the absence of a helper virus, RAAV integrates as mentioned above on a specific site on human chromosome 19 via the latency pathway. In the presence of a helper virus such as adenovirus, RAAV follows the productive pathway by replicating and forming many small RAAV virion particles. This slide illustrates the underlying technology for small-scale production of recombinant AAV. A mammalian cell line such as HEK293 human embryonic kidney cells is triply transfected by 3 plasmid vectors. 1. RAAV vector with therapeutic gene. 2. Rep and cap plasmid supplied in trans. 3. Adenovirus helper factor plasmid. Delivery of plasmids into cell can be performed by standard electroporation. In the cell factory, recombinant AAV virions are produced, the cells are lysed, RAAV particles are released, RAAV particles purified by cesium chloride procedure, and finally this results in purified RAAV ready to be delivered into the human body by intravenous, intraarterial, transdermal injection or into the human eye, retina. This is the expansion of slide number 10. Making modifications to the PAAV-ITR vector plasmid will insert varying therapeutic genes. Example, inserting the ACRP30 therapeutic gene will make the RAAV plasmid suitable for treating insulin resistance / type 2 diabetes. Making modifications to the AAV rep cap vector will alter RAAV rep capsid nature and determines cell specificity. Example, constructing a rep2/cap2 plasmid, supplying it in trans is important for treatment in insulin resistance / type 2 diabetes. The type of the rep/cap plasmid supplied in trans during the triple transfection will dictate the nature of the RAAV viral capsid and thereby dictate AAV's ability to target certain cell types, cell specificity. For example, the REP2/cap4 plasmid will make RAAV specific for application in human diseases in the eye, retina. This slide illustrates the modification to be made within the AAV cap gene so that the RAAV capsid is able to evade antibody detection after RAAV is delivered into the human body. If the antibodies were to detect RAAV, they would neutralize it immediately, thereby making the therapy ineffective. After production of RAAV virions by triple transfection, small scale, or using bioreactors large scale, it is quantified and then delivered in appropriate dosages into the human body for human gene therapy. One RAV application is in the area of HIV/AIDS, where RAV blocks the ability of enzyme integrase to perform its action in assisting HIV nucleic acid delivery into the cell nucleus. Eventually, we would like to extend AAV technology into developing small medical devices called biosensors. They will measure the appropriate level of therapeutic RAV circulating in the human body and indicate when it is necessary to take additional RAV infusions. Thank you. I hope you enjoyed the presentation. Please visit the website http://www.sjontheraputics.com and http://www.virasep.com.